Portugal. I work at MARE Research Center, and I feel kind of an outsider because I won't be speaking about snakes, although there's a little bit of scorpion in my presentation. In this case, it's the black scorpionfish, which will be the motto for something that I want to show you in the end, because these are really fresh results from our lab. And so I will be able to show you those results not only on black scorpionfish. So as you know, we have a huge diversity of venoms along the animal kingdom, from cnidarians to mollusks, including the cone snail example and the blue ring octopus, and also fish like the lionfish. Oh, and we have a snake here after all. It's the sea snake that will be the only example that I have here. But the main part is that in some cases they are quite, there's a huge conservatism of venoms in some of these taxonomic groups. And in other cases, they can change a lot, including in the same individual along their lifetime or according to their diet. These are examples that you know better than me. Okay, so we are here where I was talking about my inspiration into this research area, and it was a wonderful travel to follow the work of Baldomero Oliveira and his team, not only because they were able to pick one of the essential components of venoms that are quite complex, such as those of the cone snails, but also because they had to figure out that there was calcium channels that were from the motor system of fish that had closely related cells in the pain pathway of the spinal cord of mammals. And that's the main reason why this doesn't paralyze mammals in the same way that it paralyzes fish, but in fact it blocks the pain pathway and is an effective pain control medicine. The other opportunity that I had was to have a discussion with someone that you may know, the Juan Calvete, and something that we had the opportunity to discuss, which was this twist nature to fight pathology, which is kind of an interesting area to apply the basic science that I was used to do in fish ecology. So what about fish? What do we have on fish? We have basically some seminal works also that show us that phylogeny can be used as a roadmap, and mostly the fact that fish venoms have been largely overlooked in the literature. Although we have plenty of information, it's not comparable to other taxonomic groups that you may know better. In the same sense, it's important to stress that venoms in fish are in fact a defensive mechanism and adaptation. And sometimes this brings the discussion on, is it necessary for venoms in fish to evolve as rapidly as in the case of arms race between predators and prey? Because in this particular case, they don't have to adapt to a specific predator. Usually they exist, these venoms exist, not for a specific predator, but instead to elicit pain or discomfort so that these fish can escape their potential predators. So the other thing that I noticed is that most part of the research in this species, on fish species, is focalized on stonefish and lionfish. And there's, of course, a logical reason for that, because those are the most dangerous species, the most dangerous venoms in fish that we know, including the fact that it's one of the examples where you have an antivenom produced in the case of the stonefish. So you have some reviews on fish venoms and also some ideas on how they interact with the organisms and with accidents with humans. So basically cardiovascular, neuromuscular, cytotoxic, including hemolytic and platelet lysing, inflammatory and nociceptive activities, among others. So this is not an exhaustive list. Now moving on to a part where I'm more comfortable about talking. We have in the Northeastern Atlantic, so along the European coast, we have mainly 3 groups of venomous fish, including the stingrays, the weaverfish, and also the scorpionfish. We had to let go the stingrays for several reasons. One of them was that we were not able to keep them in our fish facility because they are either too big or they include protected species that we are not allowed to keep. So if you want a fresh supply of venom, you have to keep this fish in your fish facility, and we were not able to do that except for the weaver fish and also for the scorpion fish. In terms of the places where you should not touch when you handle this fish, in case you want to know, usually people only takes care about this dorsal area, but in fact, in the case of scorpion fish, you can find it in the opercular area and also in the anal fin, and in the case of the weaver fish, also in the opercular area. So those are the places that you don't want to touch. But even in Portugal, where you have the most experienced people handling this fish, which are fishermen, usually they get stung by these creatures, and sometimes you have accidents, especially with fishermen. About the distribution of these species worldwide, so focusing now on scorpionfish and weaverfish, you can see that scorpionfish have a worldwide distribution. And it's a strange one because usually you have most of the diversity in the Indo-Pacific area. And in the case of scorpionfish, basically the most diversity is found in the Atlantic basin. About the weaverfish, they are localized along the eastern Atlantic coast, as you can see here. So it's quite a narrow distribution of the species that compose these groups. So we had to pick on some of them because we couldn't handle all weaverfish and scorpionfish. So we moved on into scorpionfish because we cannot encompass all the species that we find worldwide because we are talking about 60 plus species. So it was not with that objective of sampling all the species that you have available, but focusing on the northeastern Atlantic area, we in fact have quite some species that we can work with. And since I come from an evolutionary background, I would like to have some kind of a comparative approach to this venom study of these species. And one of the first things that we have to do when you use a map is to see if the map is accurate. And we immediately faced a problem because when you compare the phylogeny, the only phylogeny that is published on this species with the one that we were building, the results were not congruent. So either we are wrong or what is published is not right. And so we are still figuring out this situation, including more species in our phylogenetic tree and also some more molecular markers to try to evaluate these phylogenetic relationships in order to lately compare the venom diversity also. This is the way we capture the venomous fish, usually with scuba dive. Here you can see these are quite cryptic fish. Remember that they are related to the stonefish. There's one over there, and you will only be able to see him when he jumps into the hand net that I'm handling there. And the reason why we do this is because we want to collect specimens that are in perfect conditions, because if they are damaged in the fin race, we lose the venom, okay? And so we want to keep them in our fish facility for as shorter period as possible and in the best conditions that we can. So this is one of the ways that we capture them. So that you know how we collect the venom, we use quite a high-tech technique, as you can see. It's the sponge in a tube technique. And usually, as you may know, fish don't have any kind of muscular apparatus or any kind of device to inject the venom in their victims. It's just a mechanical pressure that is made on the spine that kind of pushes the venom into the flesh of the victim. And so this is the simple technique that we use to collect the venom, and with, I must say, with some pretty good results. So moving on to the results that I was talking about, I have the group that has been studying these issues is quite large. You have José Neto, which is back there, present here in this presentation. We also have Marta Araújo and Francisca that have been working mostly in the part of the phylogeny and also in the differences between populations, because we also went into that area. So trying to figure out if the venoms also change between populations and not only species, because this slide can show you the differences between species. So if you see this SDS page gel, you can see that each of these 5 species that are presented here have their own barcode. And this means that at least this fraction of the protein components of the fish are different, even between very closely related species. So this calls our attention to the fact that fish venoms are in fact very diversified, and at least in what we can see here, there are differences between species, even closely related species. We are lucky enough that our Brazilian colleagues were already working with a species from the same genus. In this case was Scorpaena plumieri in the southeastern Atlantic. So I'll call it the Brazilian species because it only exists in the other side of the Atlantic. And we will be comparing it with our own species, Scorpaena porcus, from this side of the Atlantic. They described already three proteins, one ranging about 120 kilodaltons, the other 17, the other 14, with cytolytic vasoactive activity, hemagglutination, and also some activity against the integrity of tissue, so inhibits integrin. And these were already described and are available in the literature, but even if you point out those potential similar proteins and compare with our own results, there's plenty of room still to do some research in these other areas that you can see here. And I'm assuming that they may be similar or identical, but they don't have to be. One of the things that we also were interested in to understand was that the description of accidents with this fish in Brazil was quite dramatic. So we felt that it could be something that was stronger in terms of activity of the venom compared to the Portuguese species. And in fact, if you compare the same concentration, around 4 micrograms per milliliter, you can see that you can reach about 80% of hemolysis in this case. And in this case, you can reach only with the same concentration about 50% of hemolysis. So at least in terms of activity against membranes, it seems to be rather more potent, the Brazilian species, compared to the Portuguese species here. The other thing that we wanted to know is that if we want to use these venoms to treat or to inactivate some bacteria, then you must have an idea of how much you need until you get a toxic concentration into a eukaryotic cell. In this case, we use rabbit erythrocytes, which are known to be quite sensitive. And so we reach this threshold, which is probably an overestimation of the actual toxicity. But anyway, it's a threshold that you can memorize because we will look into this a little bit later. Going directly into the ESKAPE bacteria, the name ESKAPE is an acronym so that we can know that these are basically the bacteria that are flagged with the most dangerous ones that are actually evolving in hospital environments. So they are happy changing genes and plasmids so that they can get their immunity against different drugs, mostly antibiotics. And they are, the E is for Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and several species of Enterobacter. And what we do is try to figure out if our venoms have some kind of interesting effect against all these different species of bacteria. Well, how we do it is just to incubate a Petri dish with this bacteria, and we did it with the help of colleagues of ours from the veterinary university in Portugal, which have a laboratory that is certified to work with this kind of pathogens. One of the things that you may already heard was about diabetic foot ulcers caused by Staphylococcus aureus, which is a common complication in hospitals that has no answer sometimes in terms of clinical response to this kind of complication. So we did not pick bacteria randomly. We actually went for the ones that are flagged as some of the largest threats in hospital conditions. So results. The results show that in concentrations at least above 2.6 milligram per milliliter, we have total inhibition effects at least on Staphylococcus aureus. So we have the 6 different species, and Scorpaena porcus is effective at least against one of them, and at least in concentrations above 2.6, and we tested between 2.6 and 3.3. So if you want to look into things, as I do, The aspect of the Petri dish would be something like this. So you have an halo, a transparent halo here in the places that were replicas of the samples that we used. And at least against the Staphylococcus aureus, Scorpaena porcus is quite effective. But imagine our situation. We have the bacteria, we have the laboratory, and we have the venoms. So what we want to do is to test it with other venoms from other species. And then came the surprise. And the surprise was that although no interesting results were obtained for Scorpaena madeirensis, no results for Scorpaena notata, the results that I already described you for Scorpaena porcus, and now effective against all the strains that we tested for Scorpaena scrofa. If you prefer this in a more organized fashion than just to look at the Petri dish, the results are here. And so to cut the story short, what we did is we focused our attention on Scorpaena porcus because we have it available. But then we took the opportunity to test it against other species and venom. And in fact, the most interesting one is this Scorpaena scrofa that we were not expecting. And so to wrap up, because Ronald already showed me the 3 minutes like, to wrap up, you have here, I hope I was able to convince you that there is a huge diversity of venom components in different scorpionfish species, even closely related ones. And I can tell you also that different populations of the same species, and we tested, for instance, in Scorpaena madeirensis, some individuals from the Canary Islands, the Madeira Island, and also from the Azores, and there were differences between them. Within the Scorpaena porcus and Scorpaena scrofa venoms, especially this last one, show promising results against multi-drug resistant ESKAPE bacteria, and a comparative approach did in fact provide us some clues on how to proceed and which species to choose to study immediately after we finished our work with Scorpaena porcus. Main drawbacks, dosing venom concentrations. I don't know if you noticed, but it was above, the concentration were above the level of hemolytic activity that I showed you just before. So we are still working with huge concentrations of venom, although they are in minute dilution. And also the old problem of venom thermal lability, because in fact, if you are a fish, you are in the water, the temperature is something that is quite stable. So sometimes venoms are quite sensitive to thermal changes. And so it's not easy to work with them or to store them and to work with them later. That's the reason why we made an effort to study only species that we could keep in our fish facility. And we tested only venoms that we were freshly sampling from our species. And I hope I convinced you that fish are also an interesting area of study, and I hope you have some questions to share with me. Thank you.